Hi everyone, welcome back. So I hope that the last lecture that we had on the resting membrane potential was okay for you, that you've had a chance to kind of pause, reflect a little bit on it, to get a little bit of studying done, to check out your notes. So we're going to try things a little bit differently today, and the planet behind me seems to be flashing on and off, so apologies on the green screen effects. But today we're going to actually talk about all or none, and we're going to be talking about action potentials. And this is, again, a fundamental and important part of the nervous system that I hope you get really excited about, because this is the way that your brain talks, and this is the way that we decipher what's going on in your brain, the firing of these action potentials. And over the course of the next 2 lectures, including today, we're going to take apart and dissect out what occurs during that action potential, how your brain actually tries to make it the best that it possibly can. And we'll talk about all of the different mechanisms that it uses to ensure that your brain is still one of the fastest supercomputers on the face of this planet. So if you're ready, well, let's just go ahead and dive right in. Let me start sharing my screen with you as we get back into neurophysiology and we start talking about these all or none principles in the nervous system. So as we also just get set up, again, for those of you who don't know, I am Dr. Ju. You can get a hold of me here at this email address. Please use discussions if at all you possibly can. If you have me on Microsoft Teams, also feel free to chat with me on there as well. But you can contact me on Teams or directly on email through this email address here. Today we're going to be talking about the second chapter in neurophysiology and a little bit about action potentials in this all or none in life and neurons. There's a pretty good section if you're using Silverthorne, so go ahead and read that if you wish. But I'm going to go through and talk to you using Dr. Key's notes that he usually uses during the course of the regular year. So let's just go ahead, dive right in. So today we're going to go back and revisit that question that I had left with you in the last lecture. Why this discrepancy between minus 90 millivolts due to potassium and this minus 70 that we actually go ahead and measure in these neurons. So we'll go back over the resting membrane potential. We'll talk a little bit about these equilibrium potentials again for different ions, and then we'll talk about these really cool voltage-gated sodium channels as well. And then we'll actually get into different properties of this action potential that I got so excited about at the very beginning. Hopefully you'll get excited about it as well. All right, so let's go ahead, dive right in. Go back, revisit what we talked about at the last lecture where we were looking at these resting membrane potentials. So why is this membrane potential at minus 70 when we measure it and not at the calculated value of minus 90 millivolts? So the membrane is indeed most permeable to potassium at rest. Do you remember why that actually occurred? So remember, concentration gradients, semi-permeable membrane, but for any charged particle to cross, we need proteins or channels. And at rest, these potassium ions are moving because they have channels that are called leak potassium channels. We don't have as many of these leak sodium nor leak chloride ion channels than we do the ones for potassium. So they're diffusing a lot less, but they are still diffusing. Other ions are moving in and out. And so I told you at the last lecture, if we look at the Nernst equation, it is wonderful at calculating the equilibrium values for single ions one at a time. So we can do one for sodium, we can do one for potassium, we can do one for chloride. We can't actually do it for multiple ions that might be moving across this membrane. And I've just told you that sodium, potassium, chloride, all of these are moving to more or less degrees. So we have to have another equation that can put all of this together. And these are the ions that are the major players, the ones that are moving the most across the membrane. And again, potassium is, out of all of these, the most important one at rest. And sodium and chloride have very little movement, which we call permeability across this membrane. So this actual membrane potential can be calculated from an expanded version of this that contains each of these different ions in turn that are actually capable of diffusing or moving across this ion. And we call this the Goldman equation. And so the membrane potential, or EM, can be calculated using this Goldman potential as long as we know a little bit about the concentrations of the different ions that we find outside the cell and inside the cell. Because we want to keep it consistent, and because this is a negative charge, we flip over the equation so that the chloride concentration outside the cell is divided into the chloride concentration inside the cell. Again, very, very similar to what we looked at before. So it's just an adaptation of the Nernst equation for each of these different ion species. And when we do that, we do get about minus 70 millivolts. So let's think a little bit about this. Under certain circumstances, the permeability of sodium ions can actually become the dominant ion. And in fact, it overpowers the regular movement of these potassium ions. And because of the differences in the equilibrium potentials, that means that the membrane potential of the cell, of the neuron that we're measuring, can actually change dramatically if potassium suddenly switches over to sodium being the ion that's moving much more vigorously. So these membrane properties change to make the membrane more permeable to sodium. If something happens along that road, then we can actually get net sodium movement or sodium ions moving, which we call a current. The movement of these ions, we will actually see a net change in the current. So at equilibrium, there's a net cation accumulation inside the membrane when we take a look at the sodium equilibrium potential. So the membrane potential, again, is positive inside with respect to the outside, just keeping that in mind. And when we were to look at it, we have a larger concentration gradient for sodium outside the cell than inside. So there's that concentration gradient that we talked about. At some point, though, we know that as sodium moves in, it will start to repel itself. And as it does, because more positive charges being built up, this equilibrium will come into play. So on the one hand, we have a concentration gradient that still exists, but we can't move things in any further because we have too much positive charge inside the cell, and it being a positive ion, it can't move in any further. This value, where we have this equilibrium state developing, is known as the equilibrium potential for sodium, and we can calculate it using Nernst, and it gives us a value of about plus 60 millivolts. Now again, it's totally different. The equilibrium potential for potassium was 90 millivolts. For the equilibrium potential for sodium is plus 60 millivolts. Keep that in mind for the time being. The other ion that we had in that Goldman equation is the chloride ion, and inside the cell we have several large proteins. These are trapped inside the cell. They can only get across to the outside using exocytosis. And since they tend to have negative charges associated with them, and that was on that previous slide, and apologies, I'm going to go back. We have these negatively charged protein molecules trapped inside the cell. They can only get out through exocytosis. The chloride ion actually wants to get pushed out of the cell. And as a result, because chloride tends to be repelled by the negatively charged protein molecules, the chloride ions tend to be more concentrated outside of the cell than they are inside of the cell. And again, this is primarily due to these anion proteins that exist inside of the cell. And it's not because of any active pump. Active pumps generally work to move sodium and potassium or cations, positively charged molecules only. Hope that's clear. So normally I'd stop here to ask questions. If you have any questions, please jot them down, write them on the discussions page, or bring them to the open session on Thursdays, where I'll be happy to try to answer them for you. Hope that sounds like a good plan for you. So talking about these sodium channels, to generate the signal, the membrane has to increase its conductance, the movement of these sodium ions. And that means that we have to open up a channel that is only selectively permeable to sodium ions. Do you remember any of that? Does that ring a bell for you at all? We talked about selectivity pores, and we talked about how channels are selective for specific ions. Hopefully that rings a little bit of a bell for you when we start talking about these permeabilities and selectivities. That's what I'm referring back to in lecture number 1. So this particular sodium channel is not a leak channel, so it's not open. This is voltage gated. So we also talked about voltage gated channels in lecture 1 as well. It was a subclass of channels. And under normal resting membrane potentials, this voltage gated sodium channel is actually closed. And we talked about that particular scenario in some detail. In order for this sodium channel to open, we need to depolarize the cell membrane by a specific level. And if we can reach that level, if we can depolarize it, change the polarity or change the positive charges on the inside versus outside, and we can get it more positive inside, we might be able to also change the opening and closing of this voltage-gated sodium channel. So this channel is normally closed at minus 70 millivolts, and this again referring to the voltage-gated sodium channel. And here we're also talking about that same thing. Remember that S4 domain, those little wings that we were talking about? And here we have sort of this activation gate here, excuse me, and then typically at rest, so minus 70 millivolts, this is the resting membrane potential. This is closed at rest, so it's closed. Those wings are down, so that means that the channel itself is closed. And this starts to be depolarized somehow that we are not talking about right now. We'll get to that later on in this lecture series. This channel is only open by depolarizing the membrane. So here, if you look very carefully on this diagram, this is the normal resting membrane potential, positive outside, negative inside, minus 70 negativity overall. And then if we could somehow start to make it more and more positive inside, which is what you are seeing here, and we talked about that scenario when we were looking at the last lecture of getting that S4, that wing, to start moving upwards, the normal position, then we can actually open up this gate and we can actually start to see the membrane depolarization opens up this gate more and more frequently. And so it will fully open, as you see here. Sodium ions will be able to flow in. Sodium ions carrying positive charge will allow for more positive charge to be built up here. So we'll actually see an even greater and much more rapid depolarization. However, this particular channel has this unusual feature in that it has this other little protein piece that is known as an inactivation gate. And at a certain point in time, this will close. And so even though the S4 gate is open, or that S4 positive charge is not in place, we have this inactivation gate in place. And as a result, sodium ions cannot flow through. So no sodium ions flowing through, this will start to repolarize, as we will see in a moment. And in order for this inactivation gate to be removed, I actually have to go all the way back to the level that we were at below that threshold point, and that threshold being minus 55 here. There's a magic number that we'll talk about in a moment, this minus 55, this threshold value. We actually have to go below that for this inactivation gate to be removed and to open up this channel once again. All right, so hopefully that was clear for you. So we did a really quick summary of this voltage-gated sodium channel. We talked about the reasons why it's not at minus 90 and it's at minus 70. We talked about the Goldman equation, and then we talked about some of the kinetics and some of the things that happened in this voltage-gated sodium channel. Again, a lot to cover, and there's a lot of details there as well. So if we took all of those different pieces together, we would call this phenomenon that we just looked at an action potential. And here you can see this resting membrane potential. We can see this large increase, these massive depolarization. And then we'll see this hyperpolarization that we'll get to in a few slides. And we can see that we have two different types of channels, a voltage-gated sodium channel and this other channel that I haven't mentioned to you yet that we'll get to. Two different ions, completely different ions, are moving through these two different channels. You might want to come back and make sure that you understand what's happening in this particular animation. All right, sound like a plan? Let's get going into this action potential. So here's this really cool thing. You can only produce an action potential in a cell that has a membrane that contains these voltage-gated sodium channels. Even though we have a resting membrane potential, we can only get an excitable cell if we have these voltage-gated sodium channels. There is nothing. This is like a rule of physiology where we must have voltage-gated sodium channels to get excitation. All right, so we'll talk about that a little bit more in detail later on. And this presence of this voltage-gated sodium channel is what makes a membrane excitable, in quotes, or to produce these action potentials that I'm talking about now. And in fact, sodium channels occur in very high density within these excitable membranes. Now, keep in mind, even within a neuron, and it may not make a lot of sense to you now, hopefully it'll make sense to you after lecture number 3 or lecture number 4. Even within a neuron, not all of the membrane within a neuron is excitable. So parts of the neuron don't have voltage-gated sodium channels, which means that they're not all excitable membranes. So when these voltage-gated sodium channels are open, that membrane potential actually gets very, very close to that equilibrium potential for sodium that we calculated earlier. And these channels, though, they close or inactivate very quickly. So from the time that they open to the time that they close again or inactivate, it's only about half a millisecond. And this means that they rapidly inactivate. The sodium inactivation also leaves potassium ion movement as the main current, the main ion that is moving again. And this means that the resting potential gets to be restored. This potassium movement, though, is not through those lead channels that are responsible for the resting membrane potential. These are very specialized, as you gather, voltage-gated potassium channels, and I'll show you that in the next few slides. So again, going through the action potential stepwise, when we take a look at rest, about minus 70 millivolts, a little different than what we would calculate just due to the movement of potassium ions alone and the equilibrium potential for potassium ions. We will start to see if we reach this threshold value of about minus 55 millivolts. Here it's a little bit less, but let's assume the threshold is around minus 55. We'll open up those voltage-gated sodium channels, meaning we get more sodium entry, which is what we are seeing here. Many, many more sodium ions are flowing through those voltage-gated channels. At some point, they inactivate within about half a millisecond. So if this is 1 millisecond down here at the bottom, about halfway through, or half a millisecond, these will start to close. We get no more movement of ion. There's no ion permeability for sodium anymore through this channel. It's now closed off. That door is closed. And so the membrane potential will rapidly start to come back down. But in fact, it comes back down faster than we would normally assume because we are increasing the permeability for potassium through voltage-gated potassium channels. These are also opening because of membrane depolarization. We've reached threshold. They're also voltage sensitive or voltage dependent. They're just slower, and so they've taken time to open. And now that the sodium is closed, these are opening up, causing this increase in the permeability of potassium. And instead of coming into the cell, it's actually leaving the cell in the opposite direction. I lost my hand. Okay, there it is again. Okay, awesome. So again, this kind of leads to this really interesting phenomenon that I showed you on that animation earlier of this undershoot. And this undershoot or after hyperpolarization is an important type of consideration that we'll get to in a moment. So let's go back and revisit a few things. We talked about threshold. So for a membrane at rest, the minus 70, it could start to depolarize a little, which is what I'm showing you with my laser pointer here. And if it doesn't reach threshold, it just comes back down to the resting membrane potential. It may get excited again by different input. We haven't talked about that yet, but we're getting there. It might depolarize a little bit more, but it still isn't reaching threshold. And then if we get a big enough stimulus, it will reach threshold, open up those voltage-gated sodium channels that I showed you earlier, cause that depolarization, and then it closes and then it comes back down, and then it resets itself. And we'll see that same phenomenon happening again. But here's this really interesting thing. If you look at the size of an action potential, they're always the same. So we either, once we pass threshold, we either get an action potential, which we see here, or we don't get an action potential. So we have none. So these are, we do produce all, or we don't produce none. And this is the idea of an all or none. We don't have like half an action potential. It's either we get a full action potential or we don't get an action potential, and that's that one all or none phenomenon. So if the stimulus on the neuron was below threshold, even if we started increasing it, at the threshold, we generate an action potential. But even if that threshold stimulus, so like pressing down on your finger really, really hard or just a little bit, that's the same generation of an action potential. And that's, again, really important. They're all or none phenomenon. Again, you need a little bit of time to think about that. If you need to get up, stretch a little bit, walk around your room, go outside if the weather is nice, to think about it. It's a great time to do it, to think about what this means and what this implies. And I'm going to come back to this all or none in a few slides. So here's this other really cool thing too. Now I can have a subthreshold stimulus, similar to what I showed you before. It doesn't reach threshold, and so it just comes back down. No action potentials are being fired. And if I have it at threshold for about 10 milliseconds, I will generate an action potential that you see here. And if I had a stimulus that was actually above threshold for a longer period of time, what does that mean? Like, how does your body know that you have like a really strong stimulus that is much more than would be necessary to activate one action potential? How does it signal for like more action potential? So during this longer threshold stimulation, so as long as it is above threshold, we can actually generate multiple action potentials. So the duration of this threshold stimulus, as long as it reaches threshold, the longer it lasts, the more action potentials are generated. And that increased number is what we call the frequency of these action potentials. All right, again, another key word in our vocabulary, something for you to kind of indicate, write down, and make sure that you're comfortable with. So here again is this idea that we talked about earlier. We're going to go back and revisit it. I've shown you this diagram earlier today. And I want to go back and take a look at this in a little bit more detail. So we've generated an action potential, and we've talked about that at great length. And we know that we can inactivate this sodium channel. So we have a period of time where on a patch of membrane, these voltage-gated sodium channels are going to be mainly inactivated. So they're not going to be responsive anymore. They're all being blocked, as you are seeing here. And in fact, this sodium channel remains inactivated until it starts dropping down below threshold. And I showed you that slide earlier. And this is, again, to remove that block, we have to go all the way back down below this threshold membrane potential. But here's this really interesting phenomenon. During a period of time where we have what is known as the absolute refractory period, this absolute refractory period, none of the voltage-gated sodium channels are reset or reconfigured where this inactivation gate isn't present. So all of them are being blocked. None of the channels are free and open, and no sodium at all can flow, period. So the common analogy that is used most often is, since we look at all or none phenomenon, action potential or no action potential, if you flush a toilet and then it completely drains, you can't flush it again until that water resets. And that would be this absolute refractory period. So none of those channels are reconfigured, no action potentials at all. During the relative refractory period though, some of these channels are starting to come into this configuration down here at the bottom, where they're being reset. They are allowing some sodium ions to flow through, but not enough of them are being reconfigured. So again, even though the inactivation is about half a millisecond, this reconfiguration to where they're all reset usually is about 2 to 5 milliseconds in duration. It takes time for this sodium channel to reset, for the membrane potential to get back down below that minus 55 threshold value. Does that sound okay? Does that sound like you can understand what that basic principle is? So here's this thing, again, shown to you in a slightly different way. Hopefully this will help you to understand it a little bit, and I'm hoping that I've been somewhat clear in terms of my explanation for this. So here's this action potential where we have gone from minus 70 millivolts at resting membrane potential. We have crossed threshold, so it opens up those specialized voltage-gated sodium channels, and now we get this massive influx, but then they start to inactivate. As they inactivate, which occurs within that half a millisecond after it opens, During this period of time, this one particular channel can't open again at all, no matter what we do. You could try stimulating it, you can try like hammering it, nothing will open up this voltage gated sodium channel, and so it comes back down, and this is that absolute refractory period. So it's coming back down, as you see here. It's going back below threshold, and we talked about at threshold, if we were to try stimulating it again, it's now reset some of those inactivation gates. They're away, and this channel can open under the right stimulation. If we go and stimulate it again, we might get a very small response. Is this an action potential? You could argue that it is, but take a look at what's happening. This is during the relative refractory period, that 2 to 5 millisecond period of time, where more and more and more of these voltage-gated sodium channels are resetting in a patch of membrane. So here we might have a very small number. If we waited a little bit longer before trying to stimulate in this refractory period, because time is on the x-axis down here, so the farther along here, the more time that we've allowed for resetting to occur. If we wait even longer, we'll get an even bigger response. We have more and more of those voltage-gated sodium channels coming back online. And eventually, if we waited till the very end of the relative refractory period, we would actually see a full-sized action potential. Here is that all or none. And I'm hoping that you will understand why that all or none principle still applies. So can we generate an action potential? Yes. Is it smaller? Yes. Has it violated the all or none principle? I would argue that it hasn't. Maybe that's a topic that we can take up when we meet in person, online on Thursdays, or maybe you can come up with a better analogy than I have come up for you, but I'm hoping that it makes a little bit of sense. Here's this other really interesting phenomenon, and I actually did this in a lab one year. So I did research on hippocampal neurons. So I was growing them in a dish, and I was recording from them, and I was doing a lot of different things. You can actually block a patch of membrane or the whole membrane on a neuron, and you block it from producing an action potential, and this is what is known as a depolarization block. And the way that you can do this is you continually keep the membrane under a state of depolarization. So to generate an action potential, just as a reminder, you need to actually allow it to repolarize. But if you prevent it from repolarizing below threshold and you kept it continually depolarized all the time, those sodium channels, after they fire the first time, they'll never come back. And if you permanently depolarize it by keeping it at like plus 20 millivolts, and you can do that by keeping high potassium outside of the cell, these sodium channels will become permanently inactivated. They'll never be able to repolarize, and so no action potentials will be generated in that dish until you allow these membranes to repolarize. So to do this, again, the way that I had done this was to actually use high levels of potassium outside of the cell that would destroy the normal potassium concentration gradient. And this is what's normally responsible for keeping that membrane potential polarized to minus 70 millivolts. And you can just simply add more potassium ions in the extracellular space. And you can, again, either inject it in a living tissue, or you can just add it to the medium around cells growing in culture. And this will result in permanent voltage-gated sodium channel inactivation. That neuron, that membrane, that channel will be in an absolute refractory period as long as it stays depolarized. And it becomes inexcitable. Pretty cool. It becomes dangerous because under certain conditions, you don't want that to occur. And we'll talk about that later on. And then there's this other really interesting phenomenon, really cool phenomenon that we'll be using later on in this course as well. And this is what is known as the after hyperpolarization. And I asked you to remember that detail on that animation about 15 minutes ago. We talked about it earlier. And if you want this channel to reset and you want it to act at a later phase, you need to bring that membrane potential back down to resting or below threshold as quickly as you can. And remember, normally we're doing that with these voltage-gated potassium channels. If we have more of these voltage-gated potassium channels, in addition to these leak channels, we will actually have much more movement of potassium out to repolarize much more quickly. And as a result, the membrane potential, if we have more of these voltage-gated potassium channels, will tend to become more polarized than normal. More potassium leaves the cell than would normally happen. This makes it go below the resting membrane potential, making it more polarized than normal. And as a result, if we have these voltage-gated potassium channels and we have enough of a density of them or enough numbers of them, we will actually cause this after hyperpolarization. So a lot there to keep in mind, and there's a lot sort of to maybe think about over the last 2 lectures that we've had. And this after hyperpolarization is actually really important in terms of repolarizing the membrane, and it helps to reset it really, really quickly, and will actually drop it below the resting membrane potential of -70 all the way down to -80. There's been an enhanced permeability or an enhanced movement of those potassium ions. So here's a different type of review slide, and we're looking at this one shape here. You don't need to worry about what conductance is because we haven't really talked about it, but here's a good summary slide for you to think about being able to explain. So I don't know about you, but I often grab people in my family or just at random people out on the street, or nowadays you can't do that, but if you could, just someone who happens to be handy, or you could always dial up someone and just tell them about these voltage-gated sodium and potassium channels. And would you be able to outline what's on this slide to them? I'm hoping that you can, and I'm hoping that you love neuroscience and neurophysiology enough to be able to do that. So at this point, I'm going to stop sharing my screen with all of you because I think that was actually my last slide. And I'm going to go back, and wow, I have a nice background behind me. And I'm hoping that the last 2 lectures are ones that you can go and review at your own pace. Today we tried to switch it up a little bit by just changing up the format of how we deliver it. I'm still hoping to get some feedback from you. I need to know what works best for you. Just like learning how to do a new type of experiment, learning how to make this course enjoyable, logical, and also things that you understand well, I need your help. And please provide me with that feedback. Like, do you like having the floaty head at the side as we tried here today? Do you just want to see the slides? Do you want me to tell better jokes? I don't know very many, but I'm always willing to try. Do you want to have different things in these lectures? Today we tried to add a few more animations. I'm happy to try any of or all of these things for you. Outside of the jokes, still not so great at it. Happy to bring in my ukulele if I get enough requests for that. If I were writing this as a YouTube video, I'd say, hit that subscribe button and smash that like. But I think for today, that's enough. But I would really appreciate your feedback. As always, take good care of your synapses. Be well. Hoping that we can connect on Thursday or whenever you finish this particular lecture series. All right, so we have a few more to go in this lecture series, and then we get to start talking about special senses. And wherever you're at and whatever you're doing today, be well. All right, see you folks.